Now we're recording. So when we divide up our effectors, we know that this one segment will be skeletal muscle, and that's the so-called somatic division of the motor efferent pathway. Somatic came from information in our environment, and it requires skeletal muscle responses. Like if it's pain, we want to move away from the pain. If it's a stretch, we're going to contract, and so on. So that leaves us with the other effectors that would be described as autonomic effectors. And so that leaves us with smooth muscle, cardiac muscle, glands. We've repeated this so many times, we should all know it. Glands and adipose tissue. So we have four categories of effectors that fall into this autonomic category. So the pathway's in the same direction, it follows the same rules. There's a difference though. So what I'm going to do, I'm going to just take this arrow out and just change things a little bit. So we know that most of the integration that goes on between in the somatic system, the so-called voluntary system, goes up to the cortex. Remember, we had our somatosensory areas for receiving sensation, and then we had our primary motor areas and our premotor areas that we're going to send down motor responses to the skeletal muscle. So that would be directed in the way that we've been drawing it all along here. So we would just have our arrow come down like that. And technically, if we wanted to get a little bit technical, we could put a synapse just before we leave the spinal cord. There's a synapse. So the motor neuron that came from the primary motor cortex, it's going to go through the midbrain, through the pons. It's probably going to cross at the medulla oblongata, and then travel down the spinal cord and synapse on a motor neuron in the spinal cord and leave through that spinal nerve. Remember when we drew the gray matter? And we had our motor neurons in the anterior horn, right? Everybody remember that? And it would leave in the anterior root, right? Everybody's familiar? And we enter the spinal cord in the dorsal root. Is everybody okay with that?
Yes.
Right. So this made our spinal nerve right here a mixed nerve because it had sensory and motor fibers in it. So we're leaving motor and we're entering sensory. So that's what we've got over here. So this yellow neuron is really this neuron over here. We'll even color code it. So that's the one that's going to finish on a skeletal muscle and cause contraction if it's excited, if it's excitable. So in the autonomic nervous system, there's a bit of a difference. There's a two-neuron pathway between the spinal cord and the effectors. So it will leave the integration centers just the same way, and its synapses on a neuron will make them different because they're autonomic. In the spinal cord, which we talked about, but on its way to the effector, it synapses on another neuron before it gets to its effector. So it's described as being a two-neuron pathway from the spinal cord to the effector. And I'll show you what that looks like on a spinal cord in just a second. But we can draw it here. We can show it here. So in the first case, what we did is we had a neuron that came from the brain, and it came down, it descended to synapse on that. So this right here is what we've drawn over here. Everybody see that okay? This and this, which is the same thing. that work for everybody?
Yes.
Yeah, everybody sure about that? There's a lobe. So what we're doing here is we're looking at the brain in an anterior view. So from the primary motor cortex, we've got a neuron that's going to descend from, if this is our right cerebral hemisphere, it's going to cross over in the medulla oblongata, and it's going to travel down the spinal cord to synapse on the neuron that's in the spinal cord, and that neuron travels out and finishes on skeletal muscle. Does that see? We've done 3 different pictures of the same thing. Does that work for everybody?
Yes.
Yeah. Anybody having trouble with this? No, we're okay. Okay, so we'll come back to this in a second. So let's talk a little bit about the autonomic nervous system generally first. Maybe I jumped ahead a little early. So when we talk about the autonomic nervous system, it's important we recognize that it's mostly about change on the motor side of our pathway, because the sensory concepts are exactly the same. We're going to take sensory information, whether it's pain or temperature or touch, from organs and blood vessels and airways and intestines and things like that inside the body. And we're going to integrate that type of sensory information. But we talked about where integration in the autonomic nervous system is going to occur. Does anybody remember where that happens? All things autonomic. So we mentioned this when we were talking about functions of the hypothalamus. That the hypothalamus was responsible for integration of the autonomic nervous system. So if we have the cortex outermost, and we know that the hypothalamus is deeper in, right? It was part of the diencephalon. So this is our hypothalamus right here. So when we have sensory information, the sensory receptors, so these will be generally referred to as visceral receptors. Okay, so it's still following that 3-box pathway. So these receptors, these visceral receptors now, are going to be found on organs and internal structures like blood vessels and so on. So our visceral receptor is going to start the pathway. So it might be stretch on the muscle wall of the heart, for example. And we're going to travel through the spinal cord and stem to the hypothalamus. Whoops, I put that in the wrong place. Let's just get rid of it. So we're going to travel, but instead of going up to the cortex, integration is going to occur in the hypothalamus. And then when we go out, we leave the hypothalamus, we descend through the brainstem, and probably will cross. I say probably because we don't 100% of the time, but most of the time do cross at that medulla oblongata. So we're going to cross and then descend into the spinal cord, where we synapse on what's called a lower motor neuron. Remember when we drew the spinal cord, those autonomic motor neurons were sort of in the middle part of that gray matter, and I'll show that to you again. But when we leave the spinal cord, we're going to synapse on a neuron that's in the peripheral nervous system before we finish on smooth muscle, say the smooth muscle of the wall of an artery, or cardiac muscle. We know that cardiac muscle is going to be found where?
Heart.
Yeah, the heart wall. That's what makes your heart beat. So the pathway is a little bit different. If we followed our somatic pathway, it's going to go to the cortex. And when it leaves, it descends down, crosses at the medulla oblongata, usually, and then synapses in the spinal cord. Whoops, synapses in the spinal cord. And what are we going to put in here?
Skeletal muscle.
Yes, skeletal. Oops, why isn't that doing that? Skeletal muscle. Okay, so this picture now, we've drawn in that second neuron that's out in the peripheral nervous system. So this would be the spinal cord. So that's on the left of that dashed line I just put in is central nervous system, and on the right of it is peripheral nervous system. So we have an extra neuron in the autonomic pathway. Okay, so again, I'll show you that repeatedly through our talk on the autonomic nervous system. So when we think about this autonomic nervous system, in the somatic nervous system, the motor neurons leave the spinal cord and they synapse directly on the skeletal muscle. In the autonomic nervous system, the motor neurons leave and they synapse in a ganglia, which remember was a cluster of neurons. In the peripheral nervous system, to synapse with a second neuron, and that second neuron is going to then finish on an effector, which was the smooth muscle, and so on. So if we do a little cross section of our spinal cord, so we can sort of do a little bit of a three-dimensional, I'm going to try and get fancy here. So the first thing we'll draw in is our regular spinal. Actually, let's in here put in our little ganglion before we enter the spinal cord. So we won't worry about the other side there. Now we'll put in our gray matter. So our gray matter, remember, was the horns, the anterior horns, and the dorsal or posterior horns. And we said that we had sensory that entered, right? And we had motor. What color did we have our motor before? Green. Motor was in the anterior horn, and they left. And so our spinal cord, our spinal nerve becomes a mixed spinal nerve with sensory in and motor out. So that's heading in that direction, and this is heading in that direction, afferent and efferent. So our autonomic motor neurons, they originate in the gray matter of the spinal cord, and they're going to leave, because they're motor, they leave the same way through that ventral horn and then the ventral root. But what they're going to do is they're going to branch off. They branch off and they synapse with neurons very close to the spinal cord. Okay, and then they distribute from there. So then we'll hit an effector with the second neuron in the pathway. And here, we finished on an effector here. And in this picture, this was a receptor. Okay. So these ganglia represent clusters of neurons in the peripheral nervous system that are going to continue the autonomic signal. So if we try to organize the differences between the somatic system and the autonomic system, we can look at, we'll put somatic here, we'll make a little chart. Autonomic here. And if we look at receptors, receptors for the somatic system are described as external receptors. And those were the ones that measured external environmental factors, pain, temperature, touch in the environment. And then our receptors for the autonomic nervous system, they're inside the body. So those were our intro receptors. Okay, now we'll just make this stand out a little bit. Integration, we already talked about. Typically, integration for the somatic system is going to happen in the cortex. And we already said where integration is going to happen for autonomic. Where did we say?
In the hypothalamus?
Right, hypothalamus. And we want to talk about the effectors. So this is a way to sort of summarize the differences between the two motor divisions, the somatic motor and the autonomic motor. So when we look at effectors, we know that the effectors for the somatic system are skeletal muscle, and we know that the autonomic ones are everything else. So just for the sake of repetition, smooth muscle, cardiac muscle, glands, and adipose tissue. So we can see some obvious differences there as far as that goes. Now, when we take a look at this autonomic nervous system, just to confuse you a little bit more, the autonomic nervous system itself is divided into two divisions. So we can abbreviate the autonomic nervous system as this. Okay, so that's going to save me writing out autonomic nervous system every time. Okay, so the autonomic nervous system has two divisions. So what we just were talking about, all that stuff about two neurons leading us from the spinal cord to the effector, all that's the anatomy. That's what it looks like. Now we want to look at a little bit about how they differ from a functional point of view. So when we look at the two divisions of the autonomic nervous system, one division is called the sympathetic division of the autonomic nervous system, and the other one is called the parasympathetic division. So this is sometimes abbreviated as the SNS, and this as the PSNS. So the sympathetic division, it's really summarized as the system that's responsible for fight versus flight responses. So I'm sure that some of you have heard of this before. Some of you maybe take a little bit of psychology or whatever you might have taken in your other courses in other places, and you've talked a little bit about this. So in fight or flight, what we're talking about is mechanisms that we have at our disposal when we're challenged, whether it's a psychological stress or physical stress. And when I say a stress, that's a general term for things anywhere from fear and anger to the challenge of getting up from a sitting position. Those are all examples of stressors. And the sympathetic division would be active to help us with those sorts of things. And the parasympathetic, which is not truly always going to be opposite, but often is opposite, we can sort of summarize its functions with another little saying: rest and digest. So when we're in those kinds of modes, resting or digesting, the parasympathetic division is likely going to be dominant. So they don't turn on and off. One doesn't completely turn on or off. They're typically firing at relatively more or less rates. So when sympathetic dominates, we're probably seeing some fight or flight responses. When the parasympathetics dominate, we'll probably see more of the rest and digest. So really what we're talking about here is that in the sympathetic division, during fight or flight, we're going to be using or mobilizing energy. And in rest and digest, we're conserving or storing energy. So you can appreciate that during the sympathetic division, we're going to make lots of ATP. If you think about fight or flight, you're going to want to use your muscles. You're going to want to use your neurons. And we've talked about those cells all require a lot of ATP. So during fight or flight, we're going to burn a lot of energy. We're going to use a lot of ATP, convert it to ADP, get the energy, and create some heat, right? So that's what's going to happen under that effect. And we say, typically, these two divisions have opposing effects, but not necessarily, but not to the point where they're working, to sort of, not so much to counter the other one as to create what's called an autonomic tone. Okay, so I'll write that down for you. So the two systems, the two divisions, are often, and that means not always, opposing. And that opposition is designed to create autonomic tone. So remember way back in week 1 when we started to talk about homeostasis, and most people were hoping we'd never have to talk about homeostasis again, but here we are. Autonomic tone is really saying homeostasis. What do we say a normal heart rate is?
Is 60 to 70 beats per minute?
Okay, so good. You got a range in there. The range is probably a little more generous than that. It might be more like 60 to 90, but that's okay. 60 to 70 tells me you get that there's an upper limit and a lower limit. That's the important thing out of all of that. Well, that's the heart rate, right? So that's the cardiac muscle. So we've got the sympathetic division. In fight or flight, what do you think the sympathetic division would do to the heart rate? Make it go faster or slower?
Slower?
Faster.
Faster in fight or flight. And then the parasympathetic is going to make it go which way?
Slower.
Slower. That's good. Everybody agrees with that one. So, but we get that number that's somewhere between, say, 60 and 90. And that's a result of both divisions creating autonomic tone. So when both divisions target the same tissue, in this case, cardiac muscle, we're going to create autonomic tone. So when both divisions, that's the SNS and the PSNS, okay, so what we see is when both divisions of sympathetic and parasympathetic target the same tissue, it's called dual innervation. And the goal of dual innervation is to create autonomic tone. So in the heart, that's heart rate at rest of somewhere between 60 and 90 beats a minute. Can the heart go more than that? Absolutely. When you're exercising, your heart rate might go up to 200 beats per minute. We're not at rest anymore, and that's not really considered to be homeostasis because we've got an extreme situation going on with that exercise. But so when we're at rest and we can pick it, we know that the average heart rate is, in most people, falls somewhere between 60 and 90 beats per minute. That's because of dual innervation creating autonomic tone. The respiration rate is another example of that. Respiration rates are somewhere between 12 and 16 breaths per minute. And that's a result of dual innervation to the airways and the lungs, establishing autonomic tone. Our body temperature is plus or minus 37 degrees Celsius. Dual innervation, autonomic tone. Now, some targets don't have dual innervation. And some, we'll see, so we're going to do some examples of some of the targets for these different divisions. Okay, and we'll see that they don't all have dual innervation. Sometimes they only have one or the other. And that's fine for that particular tissue, that particular function. So if we look at the distribution of these two divisions, the sympathetic division is described as the thoracolumbar. Division, because the nerves that are part of the sympathetic arise from the spinal cord in the thoracic and in the lumbar vertebrae. And the parasympathetic is described as being craniosacral, because the nerves that end up being part of the parasympathetic originate either in the brainstem, and we talked about one of those in particular last week was the vagus nerve, or in the sacrum. So what that ends up looking like is if we drew a little schematic of the brain, and again, let's go anterior posterior. So we'll have a lobe and another lobe. So we're looking at the front. So the occipital lobe would kind of be dipped down in here, and we would end up with seeing a bit of the cerebellum, and then the brainstem. We'll do the brainstem in yellow. Okay, and then what we'll do is the spinal cord. And we know the spinal cord ends as that cauda equina. Okay, and that will represent our spinal cord. So we know that spinal nerves come off in pairs, and the autonomic nerves are going to come off as well. So if they come out of the brainstem or down here off of the sacral nerves, they're part of the parasympathetic division. If they come off the spinal cord, then they're part of the sympathetic division. And you notice I just drew short dashes for the sympathetic. So what happens is the first neuron that leaves the spinal cord is going to synapse on a neuron very close to the spinal cord. That's where the ganglion is for the sympathetic, and travel a long ways to its effector, whether that's the heart or the lungs or part of the digestive system, whatever the case might be. So we've got a short, let's call it presynaptic neuron, sympathetic, and a long postsynaptic neuron. Do you see that okay?
Yes, we can see it fine.
But well, also means you understand what I'm saying with the short presynaptic neuron and the long postsynaptic neuron? Those orange lines?
Can you just explain it one more time?
Yep, for sure. No, it's important. So we have our sympathetic neuron that's leaving the spinal cord, and it travels only a short distance before it ends up in a ganglion where there are a bunch of sympathetic neurons that are the second neuron in the peripheral pathway. So they're very close to the spinal cord. So the presynaptic neuron is short, and the postsynaptic neuron travels a long ways. It's a long one. Okay, so remember, these ones are going to be fight or flight. Okay, now, if we look at the parasympathetic ones, they leave the cranial area. So, for example, cranial nerve number 3, remember we talked about it earlier today, the oculomotor nerve. It's also got a motor branch that goes to the eyeball that will cause the eyeball to constrict. Think about rest and digest. You want less light coming into your eyeball. So you're going to close it down a little bit. So it leaves the cranium, leaves the brainstem, and it travels all the way to the eyeball before it gets synapsed with the second neuron. That in the case of the eyeball, it's right on the eyeball, but it is a second neuron. So it follows the autonomic organizational rule that there's going to be two neurons in the pathway between the spinal cord and the target, the effector. But you can see our presynaptic neuron now went a long distance and the postsynaptic neuron went a short distance. So the parasympathetics pathway is opposite that the sympathetic one was. That's what I'm hoping you see. Can you see that okay?
Yes.
Okay. So the way we would write that down is that our presynaptic neuron and over here, we're saying presynaptic and postsynaptic long distance. So our postsynaptic are these ones, and our presynaptic is up there. So again, that's just anatomy, right? That's just following the pathways. So let's say that one was cranial nerve number 3. Oculomotor went to the eyeball, caused the eyeball to constrict. But cranial nerve number 10 travels a long ways and branches to all kinds of targets. It's going to branch to the heart. It's going to branch to all kinds of segments of the digestive tract, to the lungs, salivary glands, lots of, well, not so much the salivary glands, you got the other cranial nerves for that. But that cranial nerve number 10 is a major parasympathetic nerve. Targets almost everything that the sacral branches, which will target some of the reproductive organs primarily, and up here targets stuff in the head and neck area, like the tongue and salivary glands and the eyeball. Cranial nerve 10 is going to target everything else.
Sir?
Yeah.
Would that mean that cranial nerve number 10 is a presynaptic nerve?
So cranial nerve, yes, cranial nerve number, good question, good observation. Cranial nerve number 10 is, the bulk of it is a big presynaptic neuron, but it does synapse with second neurons that would still be considered part of the vagus pathway, but they're the second neuron in the pathway.
Okay, thank you.
So let me see, I've got this picture, maybe will help us a little bit. So this is what we've just been talking about a little bit here. So let's see, this is, so you can see here, if I'll just use a black pen. So you can see short distance and then long distance. So that's sympathetic. And you see the way they've arranged these ganglia, these little blue dots are clusters of neurons. They're going to be the second order neurons leaving the spinal cord. The first ones were in the spinal cord, the second ones are outside. And that's described as a sympathetic chain of ganglia, or you'll see it described as a sympathetic ganglion chain, because they're all linked together. And in the fight or flight response, think about what you want to see happen in fight or flight. That's really a defense response. So you want all your systems that are going to benefit you in fight or flight to respond quickly. So this chain is designed to distribute and widely distribute the signals from the spinal cord. So they go through the chains, they link to one another above and below, as well as heading out to their targets. So they overlap a little bit. Now, if you look on the left of this diagram, we have this right here is describing cranial nerve number 10. This one, you can see it branch to the heart, the lungs, the digestive system and the intestines, and so on. That's cranial nerve number 10. The one up here that's going to the eyeball is cranial nerve number 3. And then we've got cranial nerve 7 for the tears. I don't know how well you can read the wording on this diagram. And 9 and lesser degree, well, salivary glands. So that's what that other one is. They're the cranial ones that are part of the parasympathetic. Parasympathetic in this picture is on the left, and sympathetic is on the right.
Sir?
Yeah.
I just had a question. So it says like it inhibits like pancreas and adrenals. Like adrenal doesn't mean adrenaline, does it? Because I thought like fight or flight, you're like filled with adrenaline.
No, adrenals are literally the name of the gland that sits on the kidney.
Oh, okay, got it. Thank you.
So it's like added to the kidney. That's where that terminology is coming from.
Got it.
But you do. But what you're thinking is not far off, because the adrenal glands produce epinephrine, and epinephrine's the other name for adrenaline. It's the more scientific name for adrenaline. But where did you see it? It inhibits stomach motility and secretion, and it inhibits the pancreas and the adrenals. Well, it inhibits the adrenals in one part of the adrenal gland. So when we start talking about the endocrine system, we'll talk about the adrenal glands. But there's outer part of the adrenal gland. That's what this is referring to. And there's an inner part. And the inner part will actually be stimulated, but the outer part will be inhibited. So that's, I think the best way for us to deal with that is just to ignore it. Because of the differences there. We'll be talking about those hormones when we get to the endocrine system. We're going to start that shortly. Okay, now let me see, I have another picture here. So this will maybe help us a little bit, I'm not sure. I don't know if that's in our way. So this is our spinal cord, that's our gray matter we're looking at right here. So you can see our gray matter, right? We're good with that. And what we're showing in here is the sensory in for the visceral receptor. So we're just showing autonomic in this picture, and we're really just showing sympathetic. So sensory in, we synapse, there's an interneuron in this diagram, and then the green is motor, autonomic motor out. And as we go out the same way we did for the somatic motor, but you see we branch. We branch down to a ganglion, there's the second neurons in the pathway. The second neurons are going to synapse and send us to up and down the chain and also back into the nerve. So we're heading back out that way. And then we're heading back out this way. So a little bit confusing, but recognize short distance to the second neuron. And those second neurons communicate with one another through the sympathetic chain. So here in our picture here, we have our chain. So our chain would be made up of these synapses here. And that chain runs from basically the thoracic and the upper part of the lumbar vertebrae. So that's why it's referred to as a thoracolumbar division. And the individual nerves that leave from the brainstem or from the sacrum are the craniosacral components. We okay?
Yes.
All right, so let's look at some of the targets. And this is probably the most important thing to get from our little discussion on the autonomic nervous system is about what we're about to talk about right now. So we can divide up a lot of different targets in the body for the autonomic nervous system. So we know that those targets have to fall under the categories of smooth muscle, cardiac muscle, glands, or adipose tissue. So we'll start by talking about some of them that are described as having dual innervation. We mentioned that term earlier. That means both divisions, sympathetic, parasympathetic, are going to target the same tissue. So we have the heart. And specifically, when we talk about the heart, we're talking about the cardiac muscle. So when the sympathetic nervous system dominates, that means we're in fight or flight. That means we're being challenged by a stress. So we expect to see that the sympathetic division will increase heart rate and increase force of contraction. Does that make sense? Everybody okay with that?
Yes, sir.
Okay, So the parasympathetic in this case is going to do just the opposite. It also innervates or supplies the heart, but it's going to slow the heart rate down. Slows the heart rate and decreases the force of contraction. So exactly the opposite, and those two together create our autonomic tone, that resting heart rate that's somewhere between 60 and 90 beats a minute. Now in the lungs, also a target that has dual innervation, but what do you think we want to do during fight or flight in our lungs?
Speed up breathing.
Yeah, so we're going to increase respirations and we're going to dilate the airways. And that kind of makes sense because if we dilate the airways, we're going to get more oxygen delivered to the lungs, which means more oxygen delivered to the different cells and tissues in the body that need them for fight or flight in order to be able to make ATP. So, and again, parasympathetics are going to decrease respiration rates and decrease airway diameter. And we create autonomic tone this way in the lungs. Now think about this. This one's tricky, but it's good. If you're on top of everything, you're going to know how to answer this question. So we're talking about fight or flight. We're talking about utilizing or mobilizing anything that will help us make more ATP. So what do you think the liver is going to do under the influence of the sympathetic nervous system? It's going to increase remember way back when we were talking about carbohydrates and proteins and lipids, we said that the liver was one tissue, was one organ that can store glucose as glycogen. But the reason that it stores glucose as glycogen is so that when we're in an increased demand for glucose, because we want to make more ATP, we want to be able to get it from that glycogen. So glycogenolysis is going to increase in the liver so that more glucose is available to the cells, because it will release it to the bloodstream and deliver it to all the different cells that are trying to make more ATP. Does that make sense?
Makes sense.
Yeah, okay. And what else do you think the liver will do along the same lines? It's another multi-syllable word. Sort of reminds you of this word, but it means something different. We would also see an increase in I might have to spell it all out before anybody guesses. Nobody's gonna guess. Gluconeogenesis. The liver also can increase gluconeogenesis. What does gluconeogenesis mean?
Production of glucose.
Producing.
Right, but what kind of glucose? From where? This is stuff we talked about.
From the diet.
Well, from things other than carbohydrates. We're making
Yeah.
glucose. So we can make new glucose from some types of amino acids and some types of fatty acids. And when we do that, it's called gluconeogenesis. And there's only a few tissues that can do it, but the liver is the major one that can do this. So that's also handy because we're trying to make more glucose available in order to make more ATP. So what about the parasympathetic division? What do you think it will cause the liver to do? The opposite to this one. It would cause the opposite to this. What would that be called? Glycogenesis. Do you guys remember these terms? I mean, you don't really remember the definitions for them, but you remember the terms?
Yes.
Yes. Okay, it is sort of important. Not even, I shouldn't even put the word sort of in there. So when we cover a topic and then we move on from that topic, you're still expected to try to retain at least some of it. And some of it, especially stuff that we start to repeat. So this is stuff you want to make sure you understand. And then it will help you understand the other parts of it. You know, if you don't understand, you know, I made the statement like during the sympathetic response, we utilize and mobilize more things for ATP production, but you don't remember that ATP is about oxygen and glucose, then me saying that doesn't help you very much. So that's why I say we're trying to build on stuff we've talked about before. And that includes understanding terms like glycogenolysis and gluconeogenesis and glycogenesis and those sorts of things. Now, in that picture we saw that the pancreas was involved. So when we look at the pancreas, again, the pancreas, we're going to talk about it when we do the endocrine system. But one of the things that the pancreas is responsible for is producing a hormone called insulin. But insulin reduces blood glucose levels. So during fight or flight, we don't want that. So we're going to see that insulin is going to be inhibited.
To do the buttons down.
Okay, I'm leaving there.
Somebody's microphone is on, and I don't think they're asking a question. If you want to ask a question, go right ahead.
So there's no
There you
opposite.
go.
There's no opposite for gluconeogenesis, or is it also glycogenesis?
No. Gluconeogenesis is making new glucose from amino acids or fatty acids. So the sympathetic will cause that to increase. And if we're in parasympathetic, that's just not going to happen.
Okay, thank you.
So the parasympathetics will do the opposite to what the sympathetics do. And so we'll see an increase in insulin release from the parasympathetic side of things. Now, the pancreas is also an organ that produces enzymes for digestion. But in fight or flight, we don't want to be eating or digesting. So we're going to decrease enzyme production. And in rest and digest, we're going to increase the enzymes. Seeing the way this dual innervation thing is working? So far, we've been pretty much just doing opposites. That's not going to hold true all the time, but for a lot of them, it does. So in the digestive system, in fight or flight, we decrease motility and we decrease secretions. In other words, we're not going to be interested in digesting food during fight or flight. And we're going to see just the opposite. We'll increase motility and increase secretions in parasympathetic, because again, parasympathetic is rest and digest. It's right in the little description, digestion. So we'd expect to increase motility and increase secretions. You know what I mean by motility, right? The movement of the food along the muscular tube between the esophagus and the large intestine. And the pupils, we're going to get dilate and constrict. Think about fight or flight. You want the pupil to dilate, let more light in, be able to see more periphery so that you can respond to different things in your field of vision. And during rest and digest, you're just trying to do the opposite. You want to sleep. Let less light in. Now, when we continue this chart, we're going to come to probably our biggest exception to the rule. And that is when we get to blood vessels, there is no innervation to blood vessels from the parasympathetic division. So there is not going to be an influence by the parasympathetic division on blood vessels. So blood vessels are either going to be able to dilate or, and in either case, it's because of the sympathetic division of the nervous system only. Now, one thing I didn't mention, it's very important. When we go back to this chart for a minute, so, when we talk about a nerve transmission, we've got a neuron that's going to synapse with another neuron. And then that's going to synapse on our effector. Okay, so this is in the spinal cord. That first neuron synapses with a second neuron. In the sympathetic nervous system, that neurotransmitter, I don't know if I want to talk to you about this, just I don't know if I'm going to mix you up here. Let me think about this for just a second here. So the neurotransmitter between the first and the second neuron in the pathway is the one we're already familiar with, acetylcholine. But the neurotransmitter that is going to activate the effector in the sympathetic division is epinephrine or norepinephrine. Okay, in the parasympathetic division, it's ACH. So when the heart is targeted by epinephrine, we increase the heart rate and increase the force of contraction. When the ACH targets the heart, parasympathetic, decreases the heart rate, decreases force of contraction. So we have a different chemical messenger, we get a different response. Okay, so the chemical messenger, that's what I forgot to talk to you about, changes between the sympathetic and the parasympathetic. So if it's sympathetic, the chemical messenger to the effector is epinephrine or norepinephrine. So we just use them we just couple them. We're not going to worry about which one is which. They're related to one another. Okay, was somebody going to ask a question?
Sorry, so I was just gonna ask if they were indicators, but then you answered it. So
So in the parasympathetic, remember, it left the spinal cord and it traveled a long ways to get to its second neuron. So it would be ACH and ACH at both synapses. So we had our effector. And this is PS and S. So we used epinephrine, norepinephrine as the chemical messenger, and ACH as the chemical messenger for the parasympathetic. So that's how we get these dual innervations like this, okay? Now I bring all this up because when we move on to blood vessels, there's no innervation from the parasympathetics, but we know the sympathetics release epinephrine or norepinephrine. So it's the same messenger, and yet some blood vessels we're going to see can constrict and some can dilate. So how is it possible that if we use the same chemical messenger, we can get both dilation and constriction? So for example, I've got blood vessels that go to skeletal muscle. During fight or flight, do you think we want to dilate those blood vessels or constrict?
Dilate?
Dilate.
Yep, dilate. Because if we dilate, we'll have more flow to those muscles, right? So for example, and what about to the heart? If we talk about blood vessels that go to the heart, in fight or flight, do we want dilate or constrict?
Dilate?
Dilate.
Yeah, of course. And what about to the lungs?
Dilate.
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 Yep, so far, same answer, right? Now, here's the trick. What about to the digestive system?
Constrict.
Exactly. We do not need extra flow to the digestive system during fight or flight. So, and another example is to the kidneys. So you can appreciate that if you're being chased by a grizzly bear on a mountain pathway in British Columbia, that the last thing you want to have to do during that chase is stop behind a bush. You can't turn around and tell the grizzly to wait just a minute. I have to just void my bladder. That's what could happen if we were continually dilating to the kidneys, because that would mean we'd filter and we'd make more urine and we would fill up that bladder, right? And that's not good during fight or flight. Does that make sense?
Yeah.
Okay. So here we see I've got three examples of dilation and two examples of constriction, but we're using the same chemical messenger. So remember how a chemical messenger has to work. What does it need on the other side of the synapse in order to cause excitation in the next neuron?
A receptor.
Yeah, it needs a receptor. So there's the clue. If we change the receptor, we can change the response. So for example, the receptors that are going to be responsible for dilation of blood vessels in the examples we're using are called beta 2 receptors. So when epinephrine binds to a beta 2 receptor, it causes dilation of the blood vessels. But when it binds to an alpha 1, so alpha signal looks like that, 1, or alpha 1, it's going to cause constriction. So there's two ways to change the message. We could change the messenger, which is epinephrine or ACH in our example, sympathetic or parasympathetic, or if we can't change the messenger, we can change the receptor. And if we change the receptor, it's possible to get a different response. In this example, we're going to get a vasoconstriction, a blood vessel constriction. So all the way down over here, there's no parasympathetic supply, but yet we get a different response, so we must have receptors. Now, I'm not going to expect you to remember these different receptors, but I do expect you to remember the concept. We can change the signal with a different messenger, like ACH instead of epinephrine, or we can change the message if we have a different receptor. You don't have to remember which the receptors are. I'm not going to ask you that. But would expect you to remember the concept. Does that make sense? Are we okay with that?
Yes, but sir, can you just repeat that sentence that you just said before?
About changing how we change the message to the second neuron or to the target?
Yes.
Okay. So there's two ways, well, there's two main ways to change the message that's intended for the target tissue. One way is to have a different neurotransmitter, like we see with the divisions of the sympathetic and the parasympathetic branches of the autonomic nervous system. Sympathetic uses epinephrine or norepinephrine. That's one messenger type. And the parasympathetic uses ACH, acetylcholine, a different messenger. So if we want to decrease heart rate, we target it with ACH. If we want to increase heart rate, we target it with epinephrine. That's one way we change the messenger. If we have the same messenger, but we want a different message, then we can change the receptor on the postsynaptic target. So in our example we've got in front of us here, we've got some beta 2 receptors, and they cause blood vessels to constrict or to dilate, and the alpha 1 receptors cause the blood vessels to constrict. It's the same messenger, different receptors, and therefore different response or message.
Okay, thank you.
Does that work? Does that work?
Yes.
Okay. Now, what about sweat glands? What do you think sweat glands do during fight or flight? They're gonna Yeah, you're going to sweat more, right? So epinephrine will target the sweat glands and cause you to sweat more. In the case of the parasympathetics, it's either you sweat more or you don't sweat much at all. So we don't need to be told not to sweat. We just have to change the circumstance. So we either get signal with epinephrine to sweat, or we don't get signal and we don't sweat. Okay. Now this one's a little trickier, but it fits. And we've talked all this time about adipose tissue being a target for the autonomic nervous system. So think about that term, gluconeogenesis. What do you think is going to happen to adipose tissue during fight or flight? We're going to have, we want to make some fatty acids available. So we're going to break down the triglycerides that are stored in adipose cells and make those fatty acids available so that we can have more gluconeogenesis. So we see an increase in lipolysis during fight or flight. You're not likely going to gain weight during fight or flight. And again, on the parasympathetic side, not too much as far as that goes. Now, does anybody know what the lacrimal glands are?
Tears?
Yeah, those are your tear ducts and your tear glands. They produce tears. So during fight or flight, we're going to, this is our one example where we don't have a sympathetic innervation to the tear ducts and the tear glands, but we do at on the parasympathetic side, and we're going to see increased secretions. So during rest and digest, you're going to increase the secretions to the lacrimal glands. Okay, so these are just some examples of how the two divisions of the autonomic nervous system target smooth muscle, that's all our blood vessels, cardiac muscle, the heart, the airways, again, smooth muscle, sweat glands, lacrimal glands. We didn't really talk directly about the salivary glands, but the salivary glands, let's put them in because some of us will recognize this. During rest and digest, we're going to see increased secretions because they're going to help with digestion. And it's less viscous. Anybody know what that term viscous means?
Thick, like less thick.
Yeah, which means more watery. So on the fight or flight side, most of us can appreciate this. You sometimes will notice that your mouth gets a little bit sticky during a stressful time, right? And you know, let's say you're playing a sport and you're getting psyched up before the sport, so your fight or flight system is in full force, and you notice that your mouth starts to get a little bit sticky. The salivary glands, they still produce some saliva, but its composition changes, so it becomes a little more sticky, increased viscosity. So in this case, the salivary glands are dual innervated, but the results are sort of variations as opposed to opposites. Does that make sense, or do you see that okay?
I have a question.
Yeah.
Don't you get like dry mouth though, like when you're
Yeah, that's
flight or
yes, exactly. So that means that the salivary production gets very thick and doesn't flow very easily,
okay.
right? So it feels very dry. It's not going to be completely dry, but it feels very dry. Right? Are you okay with that?
Yeah, thank you.
Okay, so when we talk about summarizing, let's get this out of here. Let's if we wanted to summarize the autonomic nervous system, so let's go back to our three. This now maybe makes more sense talking about it now. Maybe I shouldn't have talked about it at the very beginning. Probably just got everybody mixed up. Let's put this in. I'll put the autonomic nervous system we'll do in the purple path. Okay, and our autonomic system. Okay, so we go across this way. So we're in the central nervous system above. So the purple line is for integration, is the hypothalamus. And our pathway out is a 2-neuron pathway in the peripheral nervous system. We have the neuron that leaves the spinal cord, synapses on a second neuron before we hit the target. In the somatic, the white line is somatic, we had the neuron in the spinal cord, but that went directly for our effector. Does that make sense? Do you see that in what we talk about? Yes, no?
He was a
Yes.
no for me.
Okay. So I just put in the neurotransmitters here, right? We have two divisions. So for the neurotransmitters, we can epinephrine, norepinephrine, that was sympathetic. ACH was parasympathetic. On the somatic side, it was ACH to skeletal muscle. We know that was excitable. Okay. So for the person who said no, remember when we left, if this was our gray matter, what the heck's going on? I don't know if this is going haywire or not. Okay, so here's our gray matter in our spinal cord. Remember, we had this white matter that came around like this. Okay, white matter was tracks, ascending and descending tracks. So we said we had our somatic motor neurons that were in the ventral horn. And when they leave through the ventral root and they travel all the way directly to their target. They do not stop or synapse with anything other than the target, in this case, skeletal muscle. When the autonomic neurons that are motor, they leave and they travel out the ventral horn and then the ventral root because they're motor, so that fits. But when they get out there, they don't travel very far at all before they hit a second neuron in a ganglion. And they often synapse with a ganglion above or a ganglion below. And then they travel to their targets, which are going to be smooth muscle, cardiac muscle, glands, or adipose tissue. So these end up being the sympathetic ganglion chains up and down the thoracolumbar parts of the spinal cord. So, for example, we might hit the heart or the lungs. And our neurotransmitter at the targets here are going to be epinephrine, norepinephrine, right? And at the skeletal muscle, we all know that the neurotransmitter of the skeletal muscle is what?
ACH.
Acetylcholine.
Yeah, ACH. Okay. Now, if we were coming from the cranial part of the spinal cord, in other words, the brainstem, then we would be parasympathetic. And if we targeted the heart with ACH, parasympathetic, we'd slow down the heart rate. If we target the heart with epinephrine, we increase it. But this two neuron pathway is what we drew in purple over here. See, we left the spine, remember above this white line is central nervous system. So inside, that's the spinal cord. And then we left the spinal cord and very quickly synapsed on a second neuron before we targeted smooth muscle, cardiac muscle, adipose tissue, or glands. So all we did is, we're just keeping it as a line diagram. It's still the efferent pathway, but this efferent pathway now has an extra neuron in it if it's autonomic. Does that help the person that said they were not quite following? Is that just starting to come together?
Yes, sir. Yes, thank you.
Okay, good, good. Okay, so the white lines are our traditional pathways. 2 arrows and 3 boxes, but we still have just 2 arrows and 3 boxes, whether we're talking autonomic or somatic. But we just built in a little bit more detail, the extra neuron that's in the pathway. Other than that, the concepts are still the same. The effectors and the receptors are part of the peripheral nervous system. The integration occurs in the brain and spinal cord. If it's autonomic, the specific part of the brain is the hypothalamus. If it's somatic, the specific part of the brain is the cortex.